Alrighty then. Okay, welcome class. Welcome. Okay, so our next topic is about gram gram negative microorganisms. Okay, so let's go to the topic at hand. Hang on a sec. Just switch to my um, slideshow view. All right. So in today's video, we're going to talk about gram negative organisms that are not included in the Enterobacteriaceae family. Okay, so I think this is a misnomer, by the way. Gram-negative bacilli. So we're going to talk about gram-negative bacilli. Now, let's talk about Pseudomonas first. Pseudomonas are also known as the water bug bacillus or the water bug bacilli. Don't forget, it's the water bug bacilli. And it is a an obligate aerobe. Okay, obligate aerobe. All right. So remember our previous discussion when an organism requires oxygen, it is an oxidizer, right? So it oxidizes. It literally takes up the oxygen from the atmosphere and utilizes it for its metabolic processes. So if it's not an oxidizer, it is a fermenter. Okay, very good. Okay, very good. Hopefully you guys remember that one. We have a psychic connection. I try my best to read your minds. So anyway, let's move on to odors. Physical characteristics first, of course. Odors. The odor produced by Pseudomonas aeruginosa could be either sweet, grapefruit odor, corn taco or odor. Okay, so these are things that you might consider when your examiners might consider when they make your exams. So please be mindful of these things. Okay, and most importantly, temperature preferences. Okay, so Pseudomonas aeruginosa can grow at 37 degrees Celsius, which is basically body temperature, but unlike different members of the Pseudomonaceae family, it's the only one that is capable of growing at 42 degrees Celsius. So it's slightly thermophilic. Okay, we're not gonna lie about that. Actually, in the clinical laboratory that I worked in, one of the key tests that will determine whether or not something is Pseudomonas aeruginosa is when we grow an organism in our Campi incubator. Campi agar or Campi incubator or Campylobacter incubator, because we have a separate incubator for Campylobacter species. So if the analyzer, so for example, you run it in an auto analyzer, has some sort of what you call low discrimination, meaning that the organism in question has confusing result or variable results, you need to perform follow up manual testing. So in the clinical laboratory that I work in, once a result comes out as low discrim and I notice that it is it might be presumptive of Pseudomonas, immediately I would subculture it, and I would leave the culture media in an incubator that is above 37 degrees Celsius, because more or less, if the organism grows on that particular temperature range, it's Pseudomonas. Okay, so yeah. Now we have two ways to differentiate Pseudomonas from the members of the Enterobacteriaceae family. Again, as I mentioned before, it's an oxidizer, therefore it has an oxidase enzyme, and of course the TSI, okay, triple sugar iron agar, is K over K, so alkaline over alkaline, meaning it's non-fermentative, meaning it does not cleave ammonia from it does not cleave acetic acid from the breakdown of urea to ammonia. Okay, so that's something that you need to think about when we're talking about fermentation. Okay, so basic fermentation biochemistry. Now, we have pigments for Pseudomonas aeruginosa. There's four of them: pyocyanin, pyoverdin, pyomelanin, and pyorubin. Okay, so obviously from the names themselves, you can deduce that cyan is blue, verdin or verde is green, melanin, what's the color of your skin? What's the pigment that brings about the brownish or blackish color of our skin? It's melanin. Okay, so obviously it's going to form black colonies or brown colonies, and pyorubin is of course red. Very rarely do we see rubin, organisms that produces pyorubin, but that's that. Okay, now let's move on to blue pigments. Now, the blue pigments of Pseudomonas aeruginosa is soluble to water and chloroform. Okay, this is just added information. They're not going to ask you about this in the exams, but it is nice to know. Okay, another thing that we need to talk about is the motility of Pseudomonas aeruginosa. So Pseudomonas is a motile organism. There's no doubt about that. But what type of flagella does it have is something that you need to remember. It has a monotrichous flagella. Okay, so monotrichous flagella. So among the members of, so it's not like the other members of the Enterobacteriaceae family. Some of the members have varying flagellar structures or flagellar orientations, but the members of the Pseudomonaceae family, they have a monotrichous flagella. Okay, now culture on blood agar plates show what type of hemolytic pattern for Pseudomonas aeruginosa. It is beta hemolytic. Okay, it's beta hemolytic. Now, let's talk about the pathologic determinants of Pseudomonas aeruginosa. Now, there are four pathologic determinants of Pseudomonas aeruginosa. This includes enzymes, the pili, okay, exotoxins, and hemolysins. We mentioned pili before. This may be used as what? May be used as adherence pili or reproductive pili. Okay, so bacterial conjugation. Enzymes, toxins, and hemolysins are pretty straightforward. They either break down products in the human body or they cause some sort of toxic reaction to the human body. So it depends on what happens. Okay. Now, let's talk about the enzymes first. There are two enzymes that we consider as pathologic determinants for Pseudomonas aeruginosa. Okay, it's elastasis and proteases. So elastasis is basically an analog of I would say it's synonymous to hyaluronidase, but in this case it breaks down elastasis. It breaks down elastic products in the human body. That's why majority of the time Pseudomonas is capable of growing in connective tissue, deeper in connective tissue. You'd see a lot of these organisms in wound swabs. Okay, proteases also. Hence it causes the breakdown of certain wound. It causes the breakdown of wounds. Breakdown of skin tissue, particularly, and it can go as far as the musculature. Okay, so please be mindful of these things. Now, wounds and burns infected by Pseudomonas aeruginosa is characterized by having a blue to greenish color. To be honest with you, the reason why I remember Pseudomonas aeruginosa was when I assisted in an autopsy. Okay, so in the autopsy, the patient died from the final report from the microbiology department was the blood was showing growth of gram-negative organism. Prior to the autopsy, that was the finding. Gram-negative organism after 48 hours of 24 or 48 hours of growth. I can't remember specifically, but I think it's somewhere within that range. So what we noticed was that, the pathologist who was doing the autopsy was, he asked me to come close and asked me to take a whiff of what the smell of the organ, what's the smell of the body cavity that he just opened. I mean, he didn't ask me to put my nose into the body cavity of the cadaver, but you can smell that there's a sweet smelling or wine smelling aroma wafting off from the cadaver's body. And luckily for me, I was also reviewing for my board exams back then when I was assisting the autopsy. And I remember, I mean, they asked us, what do you think is the culprit of the disease, or sorry, the septicemia? What do you think? Because there wasn't any final report yet. Immediately, what came to mind was the bluish color discoloration of the pus inside the inside that body cavity that we opened. So I think it's the upper or the thoracic cavity that the pathologist opened. And what I saw was that there's so much greenish to bluish discoloration in the pleural cavity. We didn't open it that much. They didn't expose that much, but you can see that there is color. But I can see that there was a color blue, a bluish or greenish color, and I immediately answered it. I think it's Pseudomonas based on the smell and the color. So the pathologist agreed with me, and at the end of the autopsy, when the final reports were being sent to the when the specimens were sent and the final reports were sent to the the head of the department, um the pathologist called me. You're correct. It's actually Pseudomonas aeruginosa that caused the septicemia, leading to or multiple organ failure for this particular patient. So yeah, so that's something that I would like to share with you guys. Sometimes you don't necessarily have to look at every piece of evidence in the exams. Maybe there's one or two words that might direct you to the correct answer. And I think the easiest questions in the exams usually come from the topics of Pseudomonas. Just for your thought, just pieces of advice for you guys. Okay, now let's talk about the infections associated with Pseudomonas aeruginosa. Mild otitis is known as a swimmer's ear. Okay, if there's a skin lesion, we have what you call ecthyma gangrenosus. Okay, ecthyma gangrenosus and swimmer's ear. Okay, so where do you usually get these things? Anybody here who goes and spends their time, I don't know, hot tubbing? Yeah, so when you go to a hot tub, you might get you might get ecthyma lesions. Okay, so ecthyma gangrenosus is the exact medical term, and swimmer's ear from from Pseudomonas can also be a problem for people who are frequent swimmers. Okay, now other terms for Pseudomonas malae. So Pseudomonas malae is known as what? Known as Burkholderia malae. Okay, so another terminology for you to remember. Burkholderia malae. Okay, it's also known as Glanders bacillus or Glanders bacilli. And there's also what you call Glanders disease. Glanders disease is an animal transmitted disease to humans. What animal is usually infected with the disease? So obviously we have to remember it's a disease from horses. And any infected animals, what organ is usually involved? It's usually the pulmonary tract, or it has pulmonary involvement. So Glanders disease actually is from Pseudomonas malae, also known as Burkholderia malae. It's now Glanders bacilli, and the organism is known as Glanders bacilli. Glanders disease, on the other hand, is a disease of horses from Glanders bacilli, and horses are the main target of this microorganism, and humans can get infected because they might inhale some of the particles that the horses come because that the horses that may come on them that they may come that may come their way when they get when they're caring for horses. So yeah. Now, another term for Glanders disease is known as farcy. If this is the term that you're using, it has some sort of skin involvement or cutaneous involvement. So there's farcy, there is Glanders disease. If it's farcy, more or less your cutaneous tissue has been infected. So it could be from poking yourself with in contaminated horse products or contaminated respiratory particles of horses. So for example, taking out the horse's range. There's a lot of sharp objects there, and you might scratch yourself. So farsi would be another thing that you can associate with Pseudomonas malae. Now, let's talk about Pseudomonas malae a little bit in terms of its morphology in blood agar plates. Pseudomonas malae has brown colonies in blood agar plates. Actually, it's one of the few colonies. It's one of the few microorganisms, the gram-negative microorganisms, in fact, to form brown colonies in blood agar plates. Okay. Test animal that we use to identify Pseudomonas malae is guinea pigs. And if we're doing animal inoculations on guinea pigs, you inject it in the peritoneal in the peritoneal cavity. Hence the term interperitoneal induction. Expected results from this is what you call the Strauss phenomenon. If the organism has Pseudomonas malae, if the suspected organism is Pseudomonas malae, what happens is the guinea pig's scrotum will swell after 4 days of inoculation. 1 to 2 weeks, there is testicular necrosis. Okay, so after 4 days, if it's known as Strauss, if the suspected organism that you just injected your guinea pig with, or for example, you suspected that the patient has farsi. Let's culture that organism and let's take the brown colonies and make a bacterial suspension of it, and then do animal inoculation. You could do that, right? Intraperitoneal, you inject it intraperitoneally, and you observe that there is Strauss phenomenon. So what is Strauss phenomenon? It's basically swelling of the scrotum 4 days after animal inoculation. 1 to 2 weeks after that, you're going to expect necrosis. Okay? Now, let's talk about malaidosis. It's not Pseudomonas malae, okay? It's not Pseudomonas malae. It's actually Pseudomonas pseudomalae. Okay? It's also known as the Vietnamese time bomb disease. Okay? So malaidosis. Causative agent is Pseudomonas pseudomalae. Okay? So please don't confuse the two terminologies. Pseudomonas malae causes what? It causes glanders disease, a disease of horses that includes pulmonary involvement in these horses. Glanders disease. Okay? Another term for glanders disease is farsi. If you're using this terminology, it has something to do with skin infections, particularly involving the cutaneous tissue. Okay? Now, if your organism is Pseudomonas pseudomalae, okay? Pseudomonas pseudomalae or malaidosis, the disease is malaidosis or Vietnamese time bomb disease. Okay? Now, other names for Pseudomonas maltophilia. We have Xanthomonas maltophilia and Streptophomonas maltophilia. Okay? Xanthomonas maltophilia, Streptophomonas maltophilia. What can we deduce from the names themselves? Okay? From the name of the organism, Xanthomonas, it produces yellowish colonies. Maltophilia, maltose loving. Streptophomonas, it's what? It's a streptobacilli it's a cocobacillus or chained organism because it has some sort of chains. Now, let's look at the physical characteristics. One of the things is, of course, the odor again. The odor that is produced is ammoniacal odor. So, in Tagalog, it's mapanghe, ammoniacal odor. Okay? We have another organism that's related. That is a member of the Pseudomonaceae family, but it's been classified as a different organism. It's actually Alkalinogenus fecalis. Okay. It's a member of the Alkalinogenus species, and Alkalinogenus species are oftentimes confused with Pseudomonas species. There's one test that we can use to differentiate the two. Okay? If it's a member of the Pseudomonaceae family, it will test positive with gelatin liquefaction. But if it's a member of the Alkalinogenus family, specifically the Alkalinogenus fecalis, then that's going to test negative for gelatin liquefaction. Okay? So please be mindful that members of the alkaline genus family can be oftentimes confused with Pseudomonas. And one way to differentiate it is gelatin liquefaction. Right? Now, what is the characteristic motility pattern of the member of the alkalinogenous family or alkalinogenous faecalis? It's sluggish motility. Okay, sluggish motility. How do you know? It's like an organism that moves forward, but it's slow, and it doesn't actually go from one straight line. It just wades around, but it's still moving forward. I would consider it a sluggish motility. Now, let's talk about the Vibrionaceae family. Okay, so the Vibrionaceae family, obviously, you're going to talk about the Vibrio family, and we have other microorganisms under this family, and we're going to tackle them all together in this particular discussion. Now, let's talk about the oxygen preference of Vibrio species. Vibrio species are facultative anaerobes. Okay, they're facultative anaerobes. Therefore, they have fermentation capacity. They have the capacity to ferment sugars, right? Which will be one of the things that you need to remember. All species of Vibrio are halophilic except for the following: Vibrio cholerae and Vibrio mimicus. Okay, Vibrio cholerae and Vibrio mimicus. Vibrio species are closely related with what bacterial species? It's closely related to Aeromonas species and Plesiomonas. Okay, or Plesiomonas. Okay, depending on how you, depends on what you want, but my pronunciation is Plesiomonas. Okay, now tests used to differentiate the following is string test. Okay, or what we call the Vibrio string test. Okay, the string test requires you to use the reagent sodium desoxycholate, specifically 0.5% sodium desoxycholate, and the string test will test positive for Vibrio. Aeromonas and Plesiomonas will not test positive with the string test. What is string test, by the way? How do you do it? You just use a bacterial suspension. You make a bacterial suspension. You add 0.5% sodium desoxycholate. Observe for string formation. What happens? There is going to be some sort of small coagulum, and it will form like a mucoid string. Okay, similar to your saliva when you're when you spit something, and you want to clean it up, it sticks and it has some sort of stringy consistency. Okay, now what is the test used to differentiate Vibrio species from members of the Enterobacteriaceae family? Again, because it's a facultative anaerobe, it's capable of oxidation, similar to Pseudomonas. That's the reason why I don't discuss these two groups of organisms together, because my students might confuse them with one another. That's why I always set a different discussion, even with a different set of flashcards or PowerPoint presentations, because I want you guys to remember that the next microorganisms that we're going to talk about is a little bit different because they have oxidase. So please remember, members of the Vibrio species family are different from the Enterobacteriaceae family because they are oxidase positive. Okay? Now, what type of flagella does Vibrio species have? They have monotrichous flagella. Can I describe the motility pattern? Of course, it's again one of those organisms that have a distinct motility pattern when you observe them with the hanging drop motility observation in the microscope. Okay, the motility pattern is darting motility. So we have a sluggish motility, we have darting motility, and I want you guys to remember these things. Okay, all right. Now let's move on to culture media used for Vibrio species. All right. So there's two that you need to remember. The culture media that you use for is TCBS, also known as thiosulfate bile salt sucrose. The next thing that you need to talk about is alkaline. The next thing that you need to use is, of course, your alkaline peptone water. Both of these can be used in stool samples, or sorry, both of these. If your sample is stool samples, you may be able to use these things. Okay, what's the difference between the two? Well, of course, alkaline peptone water is a broth media because its consistency is liquid. Now, whilst TCBS is agar based or solid based. Okay, now let's move on to your next slide. So again, this is selected differential media and enriched media. Okay. Again, again, those are the other two differences. By the way, I forgot to mention. We have the serotypes of Vibrio cholerae that produces choleragen. We have O1 and O139. Again, what is O? It's the somatic antigen. What makes it different? It's the biochemical. It's the biochemical structure. So it's O1. O1 is O1 and O139 are cholera serogroups or serotypes. Okay, the choleragen acts on water and electrolyte secretion. It causes the characteristic rice watery stool. What's rice watery stool in Tagalog ba, guys? It's hugas bigas. Okay? Hugas bigas, rice watery stool. Okay? Now, let's talk about the serogroups of Vibrio cholerae. In the Philippines, we have what you call the Inaba serogroup. Hikojima is found in Japan, and Ogawa serogroup are found in India. They are endemic to certain parts of Asia. Okay, Inaba, Philippines; Hikojima, Japan; Ogawa, India. Sir, why are there Japanese names? Well, these are the names of the people who actually discovered it. Okay, so yeah, please remember these things. Of course, you may be asked in the exams, why is it important? Because sometimes Vibrio can just sit by and they would remain dormant in the environment, and then they would sometimes bloom. We call it blooming. But the scientific term for this, or the epidemiologic term for this, is an outbreak. Okay, somebody would be infected. Fecal matter fecal matter would be the fecal matter could result to cross contamination of certain things, and could reach the water. It could reach the water from could reach the water sources that's usually for drinking, and then everything goes to panic mode when an endemic when there's some sort of epidemic. Okay, so yeah. Now, let's talk about the biotypes. We have two types, by the way, classical and the Eltor biotypes. And each biotype has different tests to differentiate them. We can use the red cell hemolysis. We can use Voges Pro scour. We can use polymyxin B susceptibility test. And we can use agglutination with chicken RBCs. Okay? These are the tests that you use to differentiate it. Now, what's the name of another name for Vibrio cholerae? Vibrio cholerae is also known as the coma bacillus, and the gram stain morphology is fish in a stream. Traditionally, this is how ancient microbiologists, the non-contemporary version of identifying Vibrio cholerae is through gram stain, of course. But of course, we have automated devices now to not devices. A device is a little bit smaller in my taste. I would say an analyzer. We have automated analyzers now that can point us to the right direction, whether an organism is Vibrio cholerae. So, but this is something that you might encounter in your exams. Okay, so it's coma bacillus, and the gram stain morphology is a fish in a stream. Okay. Indicate the results of each test for the biotype of each version of Vibrio cholerae. So for classical biotypes, we have red cell hemolysis. It's actually positive for Eltor. Everything is positive for Eltor, even the polymyxin resistance test. Sorry. No. My mistake. Polymyxin is the only one that would test positive for the classical biotype. Well, the Eltor biotype will test positive for everything except for polymyxin B. Okay, this is something that you have to remember. These are one of the things that you need to remember. Okay, so red cell hemolysis, negative, positive. VP, negative, positive as well. Polymyxin B, it's the classical one is susceptible, whilst the Eltor type is resistant. And agglutination with chicken RBC, it's again the same results, negative and positive for classical and biotype respectively. Okay, Now, let's talk about sucrose fermentation. So I remember earlier that it's a facultative anaerobe, therefore it's capable of oxidation as well as fermentation. Okay, so sucrose fermenter produces a yellowish colony formation in TCBS, while a non-sucrose fermenter produces green colonies. Now, would which of the following now the following are basically the what do you call this? A summary of your sucrose fermenters. Okay, sucrose fermenters include your Vibrio cholerae and Vibrio alginolyticus, while non-sucrose fermenters include Parahemolyticus, Vibrio mimicus, and Vibrio vulnificus. Okay, so when you see green colonies in TCBS, expect that it's colored. Sorry, sorry. When you see green colonies, expect that it is going to be green, and it's Parahemolyticus and Vibrio mimicus and vulnificus. If it's yellow, okay, it's a sucrose fermenter. Okay, clear? So Vibrio is usually going to form yellow colonies in TCBS, thiosulfate, bile salt, sucrose. Now, let's talk about wound and ear infections related to marine environments caused by species of Vibrio cholerae. So, usually it's caused by Parahemolyticus, okay, and Mimicus and Vulnificus. So, yeah. Okay, so I think this one got lost in translation. Alginolyticus, sorry. Okay, now Vibrio species associated with gastroenteritis, uh, Vibrio parahemolyticus. The infection is caused primarily by ingestion of contaminated seafoods. Okay, so be mindful of these things. So Vibrio parahemolyticus. Now, we're going to talk about septicemia associated from marine environments, Vibrio vulnificus. Okay? So, septicemia associated with... So memorize mo pa siya. Yeah. Actually, my report is, of course, about Vibrio, and up to this day, I still remember it as if it was yesterday. All right. Now, what modified blood agar plates can be used or is used to demonstrate or to isolate Vibrio parahemolyticus? We use what we call Wagatsuma blood agar. And the phenomenon that you're going to observe is the Kanagawa phenomenon. And what's the description for the Kanagawa phenomenon? Basically, for non-human sources, it becomes a non-hemolytic on WBAP or Wagatsuma blood agar plates. Okay? But if it came from a human source, whether it be stool or wound sample, okay, it causes hemolysis. Okay? So that is your Kanagawa phenomenon. If the organism comes from non-human sources, maybe it again, you can isolate what parahemolyticus from where. From parahemolyticus can be isolated from marine environments, right? Salt water. Yeah. So we can observe this by getting contaminated water, right? A sample of contaminated water, and will not produce hemolytic activity on Wagatsuma blood agar plates. If it came from a human source, this will result to production of hemolytic pattern on Wagatsuma blood agar. Okay, now let's talk about TTGA. TTGA is not a band. Okay, it stands for tellurite taurochlorate. Sorry, not taurochlorate. Again, let me repeat that. Tellurite taurocholate gelatin agar. Okay, the appearance of the colonies will have cloudy zones of hemolysis, and yeah. So if it's Vibrio cholerae, you'll see this type of appearance. Okay, now what is the content of Dane's medium? Dane's medium is actually alkaline peptone water, and it can survive. And organisms like Vibrio cholerae can survive in this media for 6 to 8 hours. Okay. Now, L-Tor biotypes of Vibrio cholerae produce colonies, yellow colonies on TCBS, but turn to what color after extended periods of incubation? It turned to green. So imagine you confirm that, oh yeah, I have Vibrio cholerae. It's yellow. Just it means that it's a sucrose fermenter. But if you have an L-Tor biotype, okay, with it would cause a color change, another a secondary color change, in the after extended period of incubation. So that's one of the main differences, colonial morphology differences of L-Tor biotypes with the classical biotypes. Okay, now let's talk about the biochemical tests for Vibrio cholerae. Reagents that we use for this one is biochemical tests used for the identification of Vibrio cholerae, nitrosoindole test or cholera red test. I think this is popular when there was an epidemic, I think in the Philippines. Epidemic. An outbreak rather. The reagents that we use is peptone water, of course. It's going to be important. You have nitrate and concentrated sulfuric acid. The positive result will be a red color, and the main principle of this one is basically if your organism has Vibrio, if the test organism that you have or the organism in question is is Vibrio cholerae, it would produce nitrosoindole. Okay, therefore it will turn the color indicator red. Now let's talk about animal inoculation again. Again, sir, we have. Why do we always have animal inoculation? Well, basically, that's part of microbiology. So please, don't forget that your animal inoculation, when you're doing animal inoculation tests for Vibrio cholera, you need to use an immunized guinea pig. Okay, whether it's passive immunity or active immunity, it doesn't really matter. But what you observe is Pfeiffer's phenomenon. Pfeiffer's phenomenon is basically when even though the organism is capable, even though the guinea pigs are immunized with Vibrio cholerae, they would still die. That's actually Pfeiffer's phenomenon. All right. Now, what is the test used to demonstrate the soluble hemolysis of L-Tor biotypes of Vibrio cholerae? We use Greig's test. Okay, we use Greig's test, and the type of blood that we use in Greig's test is sheep's blood. Okay, we have also prophylaxis. So we're in the treatment CDT. It's actually a combination of drugs. Cholera, drug, cholera. I can't remember what CDT is, but let me just Google it for you guys. CDT, CDT, cholera. CDT stands for prophylaxis. Oh, it's a placebo test. So let's just omit this from from our discussion. Sorry. Now I remember. Yeah. It's cytolethal distending toxin. Okay, so it's cytolethal distending toxin from Campylobacter jejuni. Okay, so it's a CDT. It's actually known as a cholera prophylaxis. Okay, so you take one from one of the related members of Vibrio, and then we inject it to somebody, and that's basically a form of prophylaxis, prevention of cholera back in the day. Okay, now let's move on to our wait. What happened to my slideshow? Hopefully, my slideshow is showing there in our So CDT. Now, let's talk about Aeromonas species. So Aeromonas has a specific hemolytic pattern also. It's beta hemolytic. Okay. Like most members of the Vibrionaceae family or the Vibrio family, it is isolated in what habitats? Marine environments, particularly salt water. Okay. The animal that has been reported to transmit Aeromonas is leech. Okay, so leeches is a leech. Linta in Tagalog. All right. Now, characteristic flagella of Aeromonas is that it has a polar flagella. Okay, meaning it's monotrachous as well. Gastroenteritis associated with Aeromonas closely resemble that of shigellosis. Okay, so this is turning into a pattern. So a lot of the times we have enterotoxicogenic E. coli has what you call the Shiga like toxin. This time it has in gastroenteritis it's a Shiga like gastroenteritis, which is basically Aeromonas. Okay, next, Pleishiomonas is basically another member, but we don't have that much data from previous exams. So I would rather just you guys understand that it's a member also of family and Vibrionaceae. Now, let's talk about Campylobacter. Campylobacter is an organism that is known to be microaerophilic, hence the oxygen preference is known as microaerophilic environment. Please do note the oxygen requirements of a microaerophilic organisms. I've already discussed it in our previous discussion. I just want you guys to remember the microaerophilic environment and how do we do a microaerophilic gas pack system. Okay? Now, let's talk about the motility pattern of Campylobacter species. It has a corkscrew motility or a dashing motility. Okay? And a seagull wing appearance on Gramstein is most likely associated to what species of Campylobacter? It is Campylobacter jejuni. Okay, and culture or the organism is done on what culture media? Obviously, it's Campi agar. Okay, from the name itself, or you could use Kiros media. Okay, doesn't really matter. Campi pack is actually a set. Campi pack is like your Campylobacter jejuni starter kit. It has the gas pack or the jar itself, including the microaerophilic packets. Okay. Now, let's talk about the mode of transmission of Campylobacter jejuni. There's two ways you can get Campylobacter jejuni. As I mentioned before, it is a member of the Vibrionaceae family, so it's capable of living in water. So ingestion of contaminated water or ingestion of contaminated poultry. Okay. Now, the mode of transmission can be familiar. It's almost familiar to that of the answer to the second bullet is basically to salmonella. Okay, so contaminated poultry products. Okay, so this could include chicken or eggs or any other chicken products that you might or poultry products that you might be a member you might be ingesting. Okay, so yeah, notable biochemical characteristics. There are three things. For Amanda, please don't forget CHO. Okay, what's CHO, sir? Catalase, high purate hydrolysis, and oxidase. So you might see this. Actually, in my board exam, I remember there was a question like this. I could have, but the case presentation points to Campylobacter species. So I remember the examiner. One of the questions in the exam gave me this particular biochemical analysis or biochemical findings, but it was so easy to answer Campylobacter jejuni because one of the key words that I read was microaerophilic, a microaerophilic condition. Yeah, it's an organism that grew in microaerophilic conditions. Now, let's talk about pathologies. For Campylobacter jejuni, as I mentioned before, it's been associated mostly gastrointestinal disorders, gastroenteritis, diarrhea, abdominal pain. For Campylobacter fetus, it's quite different. It's associated to septicemia in kids or neonates, to be specific, fetus. Okay. Campylobacter coli, enteritis. Okay. right? Now, optimum temperature growth for, or we could do optimal temperature for the growth of Campylobacter species, 42 to 43 degrees Celsius. Remember what I just told you guys a couple of minutes ago? I have to go to the Campylobacter incubator to differentiate salmonella, to differentiate other members of Pseudomonas with Pseudomonas aeruginosa. Yeah, so that's one of the things that I do sometimes in the lab to avoid low discriminatory organisms like Pseudomonas. Okay, now let's talk about Helicobacter species. Okay, pathologic conditions include peptic ulcers, and the former name for this one is Campylobacter pylori. Okay, but it has been classified as Helicobacter because it has a different genetic makeup. Yeah, I think it has something to do with the genetics. All right, now ulcerations in the stomach caused by H. pylori is due to its ability to convert acid pH to an alkaline pH. This is because the organism is able to produce what enzyme? It's actually urease. Okay, so it's one of the reasons why you get gastric ulcerations from being infected by Helicobacter pylori. All right. Now, the specimen that we use to identify Helicobacter pylori via histopathology is through stomach biopsies. Okay, that's the direct and the gold standard for identifying or isolating Helicobacter species. You can use other tests if you read about if you studied about analysis of urine and body fluids. You have your urea breath tests or your H. pylori stool analysis or stool H. pylori stool antigen test. So yeah, but for the purpose of microbiology, stomach biopsies are the gold standard samples that you need to have. Now let's talk about the flagellar structure of Helicobacter pylori. It actually has 4 to 6 polar flagella. Okay, and it's located all over the body of the microorganism, actually of the bacilli. Now, let's move on to Haemophilus. I mentioned Haemophilus in our previous discussion. But yeah, let's start with the oxygen preference of Haemophilus species. These are organisms that are facultative anaerobes, and they need to grow at a temperature preference, or sorry, at the temperature range of 35 to 37 degrees Celsius. They are one of the organisms that you would consider as members of the Hasek group. Okay, Haemophilus is one of them. Okay, these are fastidious organisms that require certain nutritional requirements. Certain nutritional requirements include the following: delta ALA, delta aminolevulinic acid, X factor, and V factor. So please remember the other names of X factor and V factor. Right? I've already discussed it on the previous chapters. Please remember your favorite. Okay, remember your not your favorite. Remember our previous discussions. Delta ALA and Delta ALA is what? It's found in hemoglobin, right? It's a precursor molecule in hemoglobin. So yeah, you might see this in your discussion when you're talking about hemoglobin synthesis. All right. Now, this is the molecule that is a precursor molecule of hemine biosynthesis. I mentioned that earlier. Delta ALA, Haemophilus species has Haemophilus species that has this are required considered to be hemine dependent or X factor dependent. Right? Hemine dependent or X factor dependent. Now, what are the other names for the factors required by Haemophilus? X factor is known also as hemine or porphyrin. Your V factor is also known as NAD or the heat labile version. Okay? Heat stable is hemine or porphyrin. Why is it stable? Because you've already degraded the hemoglobin molecule. There's nothing else you can do. Therefore, even with extended heating, you might still have remnants of X factor. But if you heat a particular sample, a particular culture media that is enriched with V factor, you wouldn't be able to grow the organisms there because it's heat labile. Okay? Now, Haemophilus species that are X factor independent, you have a lot. There's four. X factor independent, meaning it can grow without X factor. Haemophilus parainfluenzae, Haemophilus parahemolyticus, Haemophilus aprophyticus, and Haemophilus paraaprophyticus. So just remember, if it's an X factor independent Haemophilus species, it includes aprophyticus and everything with para. Everything with para. It's parainfluenzae, parahemolyticus, and paraaprophyticus. See? Very easy to remember, right, Amanda? Just remember everything with para except for Afrophyticus. Okay? Afrophyticus. Okay? Now, Haemophilus species that are factor V dependent, or sorry, factor V independent, meaning they can grow without factor V. There's Haemophilus ducreyi and Haemophilus aprophyllus. What did you notice? Haemophilus aprophyllus is capable of growing in both in the absence of X and V factor, right? Wow. Okay. Now, what are the sources of factor X and factor V? Factor X comes from hemoglobin or degraded hemoglobin or denatured hemoglobin, depending on your persuasion. But you gotta remember that factor B comes from microorganisms, specifically Staph aureus. It could also come from yeast extract. It could also come from potato extracts. Now, microorganisms that produces factor B include Staph aureus, pneumococcus, and Neisseria. In order for you to demonstrate Haemophilus satellitism or bacterial satellitism, you just streak Staph aureus, a control organism of Staph aureus, in the culture media along with your Haemophilus species, and you'll see that they would satellite the colonies of Staph aureus. Okay, now let's talk about beta hemolytic Haemophilus species. There's two beta hemolytic organisms of under the Haemophilus family. Of course, this is your Haemophilus hemolyticus and parahemolyticus. Haemophilus hemolyticus and Haemophilus parahemolyticus. So again, for Amanda, when you hear the hemolyticus and parahemolyticus, means that it is hemolytic. Okay? Another name for Haemophilus influenzae is known as Pfeiffer's bacilli, not to be confused with Pfeiffer's phenomenon. Right? What is Pfeiffer's phenomenon? You need to infect what? You need to infect an immunized guinea pig with what? Vibrio cholera, and then it will not survive, right? But Pfeiffer's bacilli is different. It's Haemophilus influenzae. The general morphology is, of course, dewdrop colonies that may satellite Staph aureus. Okay, there are also serotypes of Haemophilus influenzae, but the most predominant or the most frequently encountered in the clinical laboratory among a survey conducted was that it was serotype B, not V, B, as in baboy, as in bakla. Okay? Now, let's move on to the pathologic determinants of Haemophilus influenzae. The most important pathologic determinant of Haemophilus influenzae is, of course, the capsule. The specimens that may be used to isolate Haemophilus influenzae include the following: blood, CSF, pus, and nasopharyngeal swab. Now, Haemophilus influenzae can be a cause of meningitis in children at what age group? 5 and below. Okay? Culture media that may be used to isolate. That's supposed to be isolate, okay? Haemophilus influenzae. You can use blood agar plates, of course, chocolate agar plates, Leventhal agar, Fieldis medium, or Fieldis agar, and the bacitracin blood agar plates. Okay? Now, characteristic odor produced by Haemophilus influenzae is the mousy odor. Okay? It's quite common to see this type of descriptions. You might remember these things. Please, please, please. You might encounter these things in your exam. So please remember the characteristics. Now, what is observed when Haemophilus influenzae is inoculated in a media with a factor B producing organism? As I mentioned before, it's satellitism or bacterial satellitism. Now, Koch-Weeks bacillus is also known as Haemophilus aegypti. Okay, this is an organism that closely resembles what biotype? It closely resembles Haemophilus influenza or influenzae biotype 3. Okay. Koch-Weeks bacillus is the causative agent, of course, of pink eye. Again, Haemophilus aegyptus. Okay. Now, Haemophilus ducreyi is the causative agent of chancroid or soft chancre. It is the smallest pathologic bacilli, by the way. Haemophilus ducreyi. Okay, chancre or chancroid. The appearance of Haemophilus ducreyi in direct microscopy, or when you examine it through direct microscopy, is the school of redfish. I think everybody here is familiar with that one. Hopefully you guys remember. All right. And basically that's it about Haemophilus. Now let's move on to Bordetella. Bordetella species, I've mentioned I think several times. Bordetella pertussis, right? Bordetella pertussis. You only have one organism that is important in this group. But most species of Bordetella requires this substance to grow. Nicotinic acid or nicotine. Okay? Nicotinic acid. All members of Bordetella species are non-motile except for Bordetella bronchoseptica. Okay? And oxygen preference of Bordetella, it is an obligate aerobe. You'll see later why. Okay? Another name for Bordetella pertussis is, of course, Bordet gengu bacillus. It is the causative agent of whooping cough, also known as pertussis, also known as the 100-day cough. Okay? The ideal specimen for the isolation of Bordetella pertussis, because it has something to do with cough, pertussis, and 100-day cough or whooping cough, it is ideal to get nasopharyngeal swabs. Okay? The pathologic determinants of Bordetella pertussis include the following: an endotoxin and an exotoxin, as well as heme agglutinins. See? It can also clump blood. Now, there are 3 stages of Bordetella pertussis or whooping cough or 100-day cough. We have a catarrhal stage, paroxysmal stage, and the convalescent stage. In the catarrhal stage, there is no symptoms, but the patient is highly infectious. In paroxysmal stage, you will see the symptomology appear, like the inspirational whoop, okay? And the convalescent stage is basically the recovery stage if the patient survives the paroxysmal stage, okay? So please be mindful, catarrhal stage, patient is asymptomatic but highly infectious. Paroxysmal stage, the patient is symptomatic with the characteristic inspirational whoop. And convalescent stage, where the organism where the patient is actually recovering, but not highly infectious anymore. All right. Now let's talk about medium. Okay, culture media for the isolation of Bordetella pertussis. So you have Bordet gengu medium, or the BGM, and the RLM, or the Regan-Low medium. Okay, so you have 2 to isolate Bordetella pertussis. Bordet gengu contains potato extract and 50% blood and glycerol. As I mentioned before, Bordetella pertussis just loves blood. Regan-Low has charcoal, cephalexin, horse blood. Preferred antibiotics includes cephalexin and amphotericin. Okay, why do we need to use antibiotics in culture media? We don't want certain microorganisms to grow, because the nasopharynx is exposed to oxygen or to air in general. There might be a lot of microorganisms that can grow there. Aside from that, the saliva and the oral cavity has normal flora, right? So we want to remove the normal flora and we want to grow the target organism that we want, right? So that's basically the reasoning why we have these types of antibiotics inside these culture media. Okay? Now, Characteristic appearance of the colonies of Bordetella pertussis. I think everybody knows the answer. Mercury droplets or pearl-like colonies. Okay? Pearl-like with the hyphen. Okay? Please correct that one. Now, kennel cough is associated with what species of Bordetella pertussis? I think I mentioned it before, but just to be sure, it's Bordetella bronchoseptica. Okay? What are the tests used to differentiate Bordetella species? You have three. Okay? You have four. Urease, motility, nitrate, and oxidase. Okay? Now, we need to differentiate the three species of Bordetella, by the way. Okay? Of medical importance. Bordetella pertussis, Bordetella parapertussis, and Bordetella bronchoseptica. Everything else will test positive for urease except for Bordetella pertussis. Okay? Motility. As I mentioned before, everything will be negative, or every member of Bordetella will have a negative motility except for bronchoseptica. We mentioned that earlier. Everything else will turn negative for nitrate except for Bordetella pertussis. And for oxidase, everybody will test positive except for Bordetella parapertussis. Now, let's move on to Francisella. Okay, Francisella, the disease caused by Francisella is not rabbit disease. Okay, a lot of my students, they would answer rabbit disease. It's actually rabbit fever. Another name for rabbit fever is tularemia. Okay, tularemia or tularensis. Tularemia. How does one acquire tularemia? Well, you can get it through direct contact, inhalation, ingestion of droplets, and of course arthropod bites. Okay? Now, when you culture tularensis, you need to use antibiotics like penicillin and cyclohexamide. Because why? Because this will grow on the skin. Usually it affects the skin and the normal flora. Again, we want to remove normal flora or contaminating substances or contaminating organisms on the isolation. Or when we're isolating, rather when we're isolating these organisms. Media of choice for for Francisella tularensis, glucosystine blood agar. Okay. Other media that may be used is PCA and CHA. Okay. Now, let's move on to Brucella species. Okay. Brucella species causes what pathologic condition? We have what you call undulant fever. Another name for undulant fever or undulant fever is Malta fever. Okay. And there are several species of Brucella. Okay. So Brucella has is the normal flora of the following organisms, of course. Brucella abortus is for cattle. Brucella militensis is for sheep and goat. Brucella suis is for pigs, and Brucella canis is for dogs. Okay. Please don't forget these things. Because why? You may encounter questions that says patient is a worker, patient is a cattle farmer, or patient is a sheep farmer, or a goat, a sheep what do you call this? Sheep shearer, meaning the job of the patient is to shear the wool, or a wool shearer, right? So a wool shearer, yeah. Okay, so yeah, that's where you get Brucella. Again, let me just repeat again: abortus in cattle, militensis in sheep and goat, suis pigs, canis dogs. I think the easiest is canis, canines. Right now, let's talk about Bang bacillus. Bang bacillus is also known as abortus. I know. It sounds salacious. I know, Mark. So please be mindful that this has nothing to do with what you're thinking. Okay, has nothing to do with banging something. It's actually just another name for Brucella abortus. Okay, culture media for Brucella is your Castaneda blood broth or milk broth. By the way, it allows the organism to survive for 3 to 4 weeks. Castaneda medium. Aside from the media of choice, we have two other media that we can use to isolate Brucella. We have Brucella agar and the Wisconsin medium. Okay. Mode of transmission of brucellosis is either direct contact of infected products, ingestion of infected animal products, or inhalation. Okay, so the animal vectors or animal reservoir hosts of this organism might have some sort of heavy infection, not heavy infection, like heavy colonization of brucella, and you accidentally inhaled something from contaminated items, you might get brucellosis. Okay? Now, tests to identify brucella. So there's only two things that you need to remember: CO2 and H2S production. Okay? Everybody would test negative for CO2 except for Brucella abortus. H2S production is quite different from for each species. For abortus, it will test positive after 4 days. For Brucella suis, it will test positive for 5 days. After 5 days, rather. Militensis and canis will test negative. Okay? Will test negative for both. How do we differentiate militensis and canis? Look at the history of the patient. Where is militensis found? Where is canis found? Right? Now, Brucella species are able to grow in the presence of what substance? Threonine and basic fuchsin. Okay? Now, we indicate the species that can grow in each substance. This is actually a funny question because this is something that was asked, that is usually asked in the board exams, but until recently they just changed this to what are the substances used to grow Brucella species. But before they said before traditional methodologies for the identification of Brucella uses which of the following would grow in threonine enriched culture media, which of the following will grow in a suspension with basic fuchsin. So those are the things that you need to remember. So I'm giving you the breakdown of the disease of that test. Threonine allows the growth of Melitensis suis and Canis Brucella organisms, and for basic fuchsin, it's Brucella abortus and Militensis. So look at the common ground here. If it grows on basic fuchsin, it's probably Brucella abortus and Militensis. But when you grow it in threonine, there's no more growth. So see, automatically the answer is Brucella abortus. If it grows on threonine, not in basic fuchsin, could be what? Could be suis or canis, right? If it grows on both, if it doesn't grow on threonine but grows on basic fuchsin, it's probably abortus. If it doesn't grow on any of them, then maybe that is not Brucella, right? Yeah, so right, see? Now, we move on to skin testing for Brucella species. So you either use brucellin or abortin. Okay? And milk media is also used for the isolation of Brucella species. Milk media is a ring probe test, by the way. Abortus ring probe test. The reagent that you use is 1 ml of milk, a drop of hematoxylin stained organism, and the positive result is actually a violet ring. Okay? And yeah, ring probe test. Yeah. So what you're doing is you're staining the organism supravitally, and they will go up the medium. Why will they go up in the media? Let's go back to oxygen preference. What's the oxygen preference of Brucella? It is what? It is CO2 negative. Most of the species are CO2 negative. Therefore, they are obligate aerobes, right? So they would go up they would go up the the milk, and they would form a ring around it. Okay? Let's move on to Pastorella species. Okay, so Pastorella multicida is what we call the type species. It's the most medically important one. Pastorella multicida is contracted from cats or dogs, and they are normal flora of these organisms' oral cavity. Okay, so they are normal flora of the oral cavity of cats and dogs. Their bites, they come from bites. Okay, Notable biochemical characteristics of Pastorella multicida is that it's oxidase positive and indole positive again. And it can ferment what sugars? Glucose and sucrose. Hemorrhagic septicemia associated in septicemia of cattle associated with Pastorella multicida is known as shipping fever. Why is it shipping fever? Sometimes the cattle will die while still shipping. So, okay. And then they found out after studying the cattle that it was actually Pastorella multicida that caused that caused the death. Hence the term shipping fever. Okay. Pastorella multicida is associated to what type of infections? Wound infections from animal bites or scratches. Remember, cats usually lick their paws when they're cleaning themselves. Sometimes, right? And you might get it from their scratches. You might get this from an animal bite. Okay? Now, Legionella species. This is something that you need to talk about. That we need to talk about in deeper, in length. What pathologic conditions can be associated from Legionella? Legionella pneumophila, Legionella mictidae, and Legionella bousmani. Right? So there's 3 species that you need to talk about. Legionnaire's disease comes from Legionella pneumophila. Legionella mictidae has Pittsburgh pneumonia, and Legionella Vigus agent of pneumonia is basically or Vigus pneumonia is Legionella bousmani. Okay, I want you to put a little bit of focus on Legionella pneumophila and Legionella mictidae. Okay, a milder form of Legionnaire's disease, or or an infection with Legionella pneumophila is Pontiac fever. Okay, now let's talk about the media of choice for Legionella pneumophila. Legionella pneumophila, we can use buffered charcoal yeast extract or BCYE. Another media that we can use is the Fili-Gorman agar. Okay, now the appearance of Legionella in BCYE is the ground glass appearance. What do you mean by ground glass, sir? Yung dinurog na salamin. Diba? Nakita niyo yung itsura ng mga yung pag nagtatayo ng, pag may nabasag na salamin, ganun yung itsura niya. Okay? Fili-Gorman produces brown colonies, okay, in this medium. Okay, now, let's move on to the best specimen for the diagnosis of Legionella pneumophila. The best sample that we can have is biopsy material. And the test used to identify organisms in this particular specimen is actually direct fluorescence, using direct fluorescent antibodies. Okay. Now, let's move on to Calimotobacterium. Sir, it's Calimotobacterium. Calimotobacterium. It's now reclassified as Klebsiella. So as of the moment, this has been changed to instead of Calimotobacterium, it's now Klebsiella species. So let's refer to it every time as Klebsiella granulomatis. Okay, so Klebsiella granulomatis is the causative agent of what disease? Granuloma inguinale or donovanosis. A characteristic morphology in a patient's pap smear, where you could see this, is of course Donovan bodies. Okay. Now, how do we describe a Donovan body? Well, it's basically a monocyte or a mononuclear cell with a gram-negative bacilli inside it, or inside a gram-negative bacilli. Okay? Now, the proposed name, I've already mentioned this, Klebsiella granulomatis. This is based on DNA hybridization, or basically it's now I think the most contemporary methodology for classifying microorganisms taxonomically is now with the 16S RNA hybridization methodologies. Okay, now let's move on with Gardnerella vaginalis. Gardnerella vaginalis is the causative agent of course, bacterial vaginosis, and the former name of Gardnerella vaginalis is Corynebacterium vaginalis or Haemophilus vaginalis. But because we have, as I mentioned before, molecular technology to identify these organisms, we classified it as a different microorganism itself, Gardnerella vaginalis. Okay? Now, describe the vaginal discharge of a patient infected with Gardnerella vaginalis. Foul-smelling, grayish vaginal discharge. Okay? Now, let's talk about the cytological test for used to identify Gardnerella vaginalis, pap smears. Okay, and what we're looking for, or what cytological feature we're looking for, is clue cells. So what's clue cells? Basically, these are groups of bacteria or gram-negative bacteria that surrounds the epithelial cells. Okay. Now we have another test, or the odor test. We have a whiff and sniff test. Basically, it's the usage of 10% KOH, and the positive result for this one is a fishy odor. Okay. Human isolation of Cardinal vaginalis is done on what medium? HBT. Basically, it stands for human blood bilayer twin medium, or tween. Sorry, not twin. Tween medium. Okay? Human blood bilayer tween media. Okay? Now, let's move on to another microorganism, Streptobacillus moniliformis. Streptobacillus moniliformis is the causative agent of rat bite fever or Haverhill disease. Okay, from the name itself, it's rat bite fever. Haverhill disease is basically eating something or eating something or ingesting something that is from milk based or contaminated milk based products. So for example, rats have been prevalent in your house, or have been invading your house, and they go to a lot of milk products around your house. Like for example, your chocolate wafers, they would step on it. They could contaminate it, and you could get Streptobacillus moniliformis via ingestion. Okay, you could also get it by getting bitten by a rat. Okay, so be mindful of this microorganism. So rat bite fever, and of course, Haverhill fever. Okay, what special indication is needed in the culture of Streptobacillus moniliformis? You need to use SPS. You must avoid the usage of SPS rather. Why? Because SPS is toxic to Streptobacillus moniliformis. What is SPS, sir? SPS stands for sulfopolyanethol sulfonate, which is part of your which is the anticoagulant that is used for blood cultures. Okay, so you need to use a different blood culture bottle that doesn't have SPS when you want to isolate Streptobacillus moniliformis. Okay, or when you're expecting Streptobacillus moniliformis. Okay, now describe the broth appearance of Streptobacillus moniliformis in thioglycolate medium. In thioglycolate, it looks like a string of pearls or puff balls. Okay, we have now Capnocytophaga species. Okay, Capnocytophaga species are organisms that have a characteristic motile pattern, motility pattern rather. It has a gliding motility pattern, so it goes wee, wee, like that. It stops and then proceeds fast and then decelerates. Okay. Now, describe the gram stain appearance of Capnocytophaga. Well, basically, Capnocytophaga is a fusiform bacteria. Okay, it's filamentous also. Right. Right. Now, let's talk about another one, Echinella corrodens. Echinella corrodens is characterized as a normal flora of the gingiva or the bowel, and the colonial morphology is that it's basically an organism that produces colonies that pits the agar. Okay, it pits the agar. It causes pitting of the agar. We have also Chromobacterium violaceum. Okay, Chromobacterium violaceum is normally found in habitats around soil and water. And the term for the pigment produced by Chromobacterium violaceum is known as violacine. Okay, violacine. Another thing that you need to talk about under the members of the HASEC group is Cardobacterium hominis. Cardiobacterium hominis is part of human body's normal flora, specifically in the respiratory tract. This organism rarely causes pathologies. However, it's been isolated in cases of patients with endocarditis. Okay? Next, we're going to move on with Bartonella species. Bartonella species has several characteristics. It is a fastidious organism. It's oxidase negative. It's best grown in blood-enriched medium, and it can grow in cell co-culture methodologies. Okay. Previously, Bartonella species were what group of organisms? They are previously members of the Rickettsiales family, or the Rickettsia family. Okay. Now, based on the geographic distribution of Bartonella bacilliformis, where can we find it? It's found in Latin America, okay, or in the Andes Mountains. The vectors that could transmit Bartonella bacilliformis include sandflies and phlebotomous flies. We're going to come back to sandflies and phlebotomous flies when we talk about when we talk about hemoflagellates. But for now, since we're in bacteriology, please be mindful that Bartonella bacilliformis can be transmitted by sandflies and phlebotomous flies. Okay? Stages of disease caused by Bartonella bacilliformis. We have two. We have acute fever and we have the chronic fever. Acute fever phase and the chronic phase. Please be mindful of the names or the nomenclature. For the acute phase, we have what you call the Oroya fever. Okay? Oroya fever. Sorry. Somebody texted me again. Terribly sorry. Okay. Alright, so it's Oroya fever. And then, hang on a sec. Let me just come back to my slideshow. Alright, so it's Oroya fever for the acute phase or the acute fever phase. It is characterized by hemolytic anemia. And for the chronic phase, it's Verruga peruana or the Peruvian wart. It's a wart-like disease. Collectively, this is known as what you call Carrion's disease. Okay? So nomenclature-wise, if you're talking about the symptomology, you refer to it as either acute phase or the chronic phase. Yeah. So everything else, or if you combine everything else, it's known as Carrion's disease. Now, the ideal specimen for the isolation of Bartonella bacilliformis includes blood. Okay, the ideal specimen is blood, not includes blood. It is blood. Okay. Now, we have another microorganism associated to the family of Bartonella. We have Bartonella quintana. It causes trench fever, which is transmitted by a vector known as which is a body louse. Okay, mga garapata. Okay. We have CSD, cat scratch disease, another species associated with Bartonella species. So it's Bartonella henselae. Okay, and Bartonella clarigidae. Okay, clarigidae. Now, IV drug users are at risk of endocarditis because of what Bartonella species? We have what you call Bartonella elizabethae. Okay, Bartonella elizabethae, and the vector for this organism is rodents. Okay. Now, we're going to end our discussion there when it comes to other gram-negative microorganisms, because we're going to continue with anaerobic gram-negative bacilli. Okay? Right? So anaerobic gram-negative bacilli. So after this one, you guys have a break. I hope we'll come back with the discussion on anaerobic gram-negative bacilli. Okay? This is just a short discussion, probably like a 20-minute discussion or an hour of a discussion. 20 minutes or 30 minutes, 40 minutes, depending on my mood. Okay? So yeah, that's basically it, and I'll see you guys after your break. All right, bye.